I think it goes without saying, and this is a theme that's been echoed in this summit, that what we'd really love to see is a future where we can treat children and adults with cancer more precisely. And the idea is that we can characterize tumor genomes or other omic aspects of childhood tumors. We can identify targets, develop new targeted therapies, with the ultimate goal of really seeing durable responses, as have now been seen in childhood ALL, in which children have BCR-ABL rearrangements with the incorporation of imatinib into their therapy. But we really face a major challenge in many childhood cancers, in that many of the targets that we've known about for years and the targets that we continue to discover are very difficult drug targets. Many of these targets are not the kinases that Charles described earlier, but they're rather transcription factors, aberrantly activated, and these really do make for difficult pharmacological targets. So we've been involved in a series of collaborative projects over the years where we've asked the following question in proof of concept. Can we take collections of pediatric cancer cell lines, or now as well primary patient material, and characterize deeply the genetic molecular features of these cell lines? Can we then perform both functional genetic with shRNA and now CRISPR screening, as well as chemical screens against these large collections of cell lines, and begin to connect mutations or molecular abnormalities with response to either a small molecule therapy or to genetic suppression of a target? My laboratory works on a number of childhood cancers, including the acute leukemias, Ewing sarcoma, and neuroblastoma. But what I'd like to briefly tell you about is a neuroblastoma project, which forms the basis of our Hyundai grant. So neuroblastoma, as you know, is the most common extracranial solid tumor in children. It's of neural crest origin, and unfortunately, high-stage disease is quite common. About half our patients present with high-stage disease. And MYC-N amplification is a genetic feature that's present in somewhere from 25 to 30% of these tumors, and this is a very poor prognostic feature. Until recently, MYC-N was thought to be intractable. It is a transcription factor. Well, we've engaged in a number of these screens of cancer cell line collections, and this was a screen that we did collaboratively with Jay Bradner's laboratory and Cyril Bennis at Mass General. And we screened a large collection of cancer cell lines defined by molecular features against a compound called JQ1. This is a new molecule. It's in a class of compounds called BET bromodomain inhibitors. And these are compounds that are epigenetic regulators. And so what we found in this screen is that N-MYC amplification was the best molecular predictor of response to these compounds. And in particular, MYC-N amplified neuroblastoma was really quite sensitive to this compound class. So a little bit more about these molecules. The BET subfamily of bromodomain proteins are epigenetic readers. These proteins bookmark DNA, and this leads to altered gene transcription. This is a very exciting topic for the pharmaceutical industry right now, and numerous pharmaceutical companies have BET bromodomain inhibitor protein inhibitors, and these programs are now really in clinical trial. And at AACR this year, there was a press release noting some very dramatic responses to these molecules in patients with AML. These compounds were reported to lead to a decrement in c-MYC expression in a subset of cancers. And so one of our first questions was whether these compounds were altering MYC-N and neuroblastoma, and we found, in fact, based on these Western blots, as you see, they had a pretty dramatic effect on MYC-N in most of the cell lines that we tested. We also then tested these compounds in collaboration with Bill Weiss's laboratory in a number of preclinical models, and this just shows response in a MYC-N amplified genetically engineered mouse model, where we saw a very dramatic response to the BET bromodomain inhibitor JQ1 with just one cycle of treatment. These tumors became Ki67 low, and we also observed cleaved caspase-3 as a marker of apoptosis, and in many of the MYC-N neuroblastoma cell lines, what we see is actually cell death as apoptosis. And so we, along with other groups, there was a report recently by GlaxoSmithKline demonstrating a very similar finding with their compound in neuroblastoma, are now quite interested in bringing this to clinical trial. And there's a clinical trial in development led by Susie Schusterman at Dana-Farber, with the idea that we really can make these discoveries, we can do preclinical testing and bring this work to clinical trial. But the work isn't done then. We really have to now take what we learn in the trial and really bring this back to the laboratory. And I think a very critical question as we're bringing these targeted therapies forward in childhood cancer is really, are we hitting the target? Because if the trial is a negative study, we don't know, is this a bad target or is this rather a bad drug? And so the focus of our project now is to bring this compound to a clinical trial and to do the critical correlative biology studies for that clinical trial. And so just very briefly, in childhood cancers, in contrast to leukemia, it's actually quite difficult. In childhood solid tumors, in contrast to childhood leukemias, it's quite difficult to serially sample tissue from the tumor. And so we've been considering alternative approaches in the context of this trial to direct serial biopsies. And so one is that we can use surrogate tissue. And so what we've been doing in the laboratory is developing a series of assays to actually measure in patients' peripheral blood whether we're hitting the target in a white blood cell population. And we're doing this both by flow cytometry for genes whose expression we know go down with the compound even in normal cells, as well as using an RNA-based assay. We're also exploring the use of circulating tumor cells, which I think really could transform how we're doing clinical trials and monitoring these patients. And so there's been a lot of work done in the adult community looking at circulating tumor cells from patients with epithelial cancers. And so collaboratively now with the Broad Institute as part of this project, we're developing an assay for looking at neuroblastoma circulating tumor cells. And we think we'll be able to use these as a way potentially to monitor minimal residual disease, as well as to look at whether we're hitting the target in these various cells in the patient. And we also imagine using these cells as enabling our ability to understand resistance mechanisms to these targeted therapies. So just to summarize, we're developing a clinical trial to test these compounds in children with solid tumors, enriching for children with neuroblastoma. We're conducting the correlative biology studies to measure target engagement in surrogate tissue, and also developing a transformative assay to harvest these circulating tumor cells and then to study them in the laboratory. And finally, we're also developing large-scale screens to identify resistance mechanisms to BET inhibitors, as these mechanisms are as yet unknown. So with that, I just want to thank everybody here for your attention, and certainly Hyundai for their amazing support. And then just to acknowledge, as I did, Jay Bradner and Bill Weiser, two of our key collaborators on that project. Thank you.